genetics. Today he'll be talking about one of my favorite topics, the predictability of influenza A evolution. John, are you okay with us recording? It's just for people from the network who might not be able to attend.
Sure, absolutely. Yeah, that's no problem.
Great. So I'll launch the recording now.
Recording in progress.
And are you okay with being interrupted with questions as you go?
Absolutely.
Okay, great. Yeah, we prefer that as well. But I'd ask everyone to mute their microphones when they're not asking a question and just to mute themselves when they are. So yeah, John, the floor is yours. Thanks very much.
Yeah, thank you very much for the invitation and for the introduction. So if if anyone has questions as I'm going along, please feel free to interrupt me as you were saying. And if you type something in the chat, I might not see it, but if someone else sees it, you can certainly call my attention to that, and I'd be happy to answer questions that way too. Yeah, so to get us started, just as you were saying, my lab is really interested in understanding evolution, especially the evolution of viruses and how they interact with the immune system. And we try to model these things and think about these things in a quantitative way. So I know for this audience here, I don't need to give you too much background on flu, but to just kind of set the stage a little bit and tell you about our perspective for thinking about this. I think one of the things that's quite interesting about studying the evolution of viruses is how rapidly they evolve, and which really allows us to see lots of evolutionary change in a short amount of time. So taking, for example, flu, we know that these error rates that the virus has when copying its genetic material can be very, very high, near the limits of mutational tolerance. The replication cycle for viruses like influenza is also very fast. So in this case, infected cells can live for on the order of 12 hours or something like that. So these sort of cycles are very, very quick. And they also have other methods for generating genetic diversity as well. So one of these things that occurs for influenza is reassortment, where I can have, in cases of co-infection with two different variants of the virus, you can have variants that are produced that have some combination of gene segments from the different variants that are different than what either purely one variant or purely the other variant, which can also increase genetic diversity in different ways and lead to the complicated evolution of these types of viruses. So all of this has consequences for our own health, which again, in this audience, I know you're very, very familiar with this topic. And I think one of the ways that this was illustrated really beautifully is this work from Trevor Bedford and collaborators looking at the evolution of flu viruses over the course of multiple decades and seeing how they've changed antigenically over time, in this case based on sort of projecting down the antigenic change based on hemagglutination inhibition assays. You can see this progressive divergence in antigenic distance between the strains of the virus. So this is why, for example, if we get flu, this year, we might be protected for a while, but then we might be susceptible to having flu again, some years in the future. So we have this process of antigenic drift, where the virus is progressively accumulating mutations in surface proteins allowed to escape from antibody responses. And the way that we think about these problems, or the things that we're interested in, is trying to understand, based on looking at data, which mutations are important mutations that appear to be really driving the evolution of the virus. So these could be ones that are involved in antigenic change or in other important functional changes. And can we use that information to try to actually predict future dynamics? And that's one that we've been thinking about for a long time. And I think finally in this work, we're starting to get some results in that direction. So I'm very excited to present this to you all. So I'll focus today in terms of actual biological applications on flu, but I want to also mention that the questions that we're thinking about are rather general, and we're not really, let's say, specifically focused on building a model in particular for flu or something like this. Some of the other viruses we've thought about are things like SARS-CoV-2 or HIV in other contexts. So we're really interested in this problem of learning from sequence data more generally and trying to understand what are mutations that are important for driving adaptation in these types of viruses, and how can we develop predictive models. So that's the kind of very broad level thing that we're interested in trying to study. But trying to make these type of predictions for influenza evolution is challenging, right? So we know that when people have infections, they develop immune memory that provides them protection against these versions of the virus that they've seen, that they previously encountered. And so even as new variants are spreading in the population, you have immunity that is growing. So it seems like the sort of landscape that you're seeing, this immune landscape, is constantly shifting. So making these types of predictions of what types of strains of the virus might be dominant in the future is something that could be challenging. In addition, if you look across populations, you have not just a uniform sort of infection history across different people, but something that's also really heterogeneous, right? So my history of vaccination and exposure to different viruses is going to be different from someone else. And so I have this very heterogeneous shifting immune landscape that can make it really challenging to think about how evolutionary dynamics should actually play out. But we're going to try to do our best anyway. And the tool that we have for this is using this incredible resource of genomic surveillance data that has been collected from labs all throughout the globe. Flu was, of course, one of the major viruses where this really began, but we saw this explode for SARS-CoV-2 and has been increasing availability for other viruses and pathogens as well in recent years. And so this can be a really amazing resource for us to follow evolutionary dynamics of these viruses and hopefully learn from them. So you can see you have actually this sort of super exponential growth in the number of virus sequences that have been collected over time. For example, here for flu and GISA, but this is happening for many other viruses as well. And often, you've probably seen these applied in things like phylogenetic analyses that are presented on NextStrain, which are really amazing ways to look at and think about these various populations. The way that we're going to use the data is a little bit different. And in particular, what we're going to try to do is we're going to look at the sort of population dynamics. So how the distribution of virus sequences is changing over time, try to learn from that to understand or try to estimate the effects of different mutations and make predictions. And I'll tell you much more about that as we go along. But that's basically the idea. Look at how these populations are changing and then use those to make at least short-term predictions about how these virus populations will evolve in the future. So that's roughly the sort of two halves or the two parts of the talk that I'm going to tell you about today. So one is sort of the more technical part. So how do we actually do this estimation of fitness effects of mutations from evolutionary dynamics? And second, I'll tell you about how we've applied this to look at influenza evolution and sort of what we found in terms of whether we can predict things like whether individual mutations will fix or be lost, and then also just generally the composition of the viral population in the future. All right, so let me tell you about the first part. So I'm going to present you with a very, very simple example to give you an idea of what we're thinking about. So you can imagine that let's say we're tracking some pathogen over time, and maybe I have two different variants. I'll call them variant A and variant B. Variant B has a mutation that distinguishes it from variant A. And at early times, I might see that most of the population, if I look at individuals with infections, this pathogen, they have infections with variant A. We have a small number with variant B. But over time, maybe I see that these infections with variant B starts to increase until at some later time they become completely dominant, taking over the viral population. So if I were to see dynamics like this, then that would likely lead me to conclude that this variant B has an advantage compared to variant A. And I could try to maybe look at how this frequency is changing over time to understand exactly how beneficial is this variant compared to the other one. But when I look at real evolutionary dynamics, it's much more complicated, right? I have lots of different mutations that I'm observing over time, and they can come together in very complicated ways, correlated with each other. And so trying to understand what the effect of any individual mutation is not so obvious. So this here in particular is showing you sort of smooth mutation frequency trajectories for hemagglutinin in H3 and 2 in humans collected over the course of about 10 years in GISAID here. So the way that we are trying to look at this data is to write down a model that incorporates the potential effects of different mutations on transmission into a model of disease transmission, and then to try to infer or estimate the effects of mutations that best explain the evolutionary history that we see, including these kinds of correlations between different mutations or linkage disequilibrium in population genetics sort of terms. So let me tell you a little bit about the model. So the model that we have is very, very simple. So we imagine that if infection is being transmitted through something like a branching process. So if I imagine that I have one person who has an infection, then they can transmit that infection on to a random number of new individuals. It could be one person in some cases, it could be two, or it could be some higher number. And in principle, this is described by some probability distribution that has an average value R. So R is our effective reproduction number. And what we include in our model is we say that this effective reproduction number is going to depend on the genotype of the virus. So it's going to depend on what variant of the virus someone has that they're infected with. And we can define sort of a variant fitness, call this WA, which I'm going to imagine multiplies this sort of baseline effective reproduction number. So we can also go in and to try to determine by the individual mutations that variant bears. So let's say, for example, I have, for this particular variant A, I have mutations, sites I and J, or particular mutations, call them I and J. Then I would say that this variant fitness is going to be get contributions from each one, additive contributions from each one of those mutations. So here, this is a very sort of simple assumption. It's kind of the simplest model that we can write down, where we're trying to incorporate the effects of different mutations into a transmission model. And then what we have is a branching process model of infection that describes how different variants can be transmitted over time. So for example, it might see dynamics that look something like this schematically, where the different colors represent different variants. And in this particular case, I might assume that the variants that are in this darker color have fitness advantage relative to the ones that are in a lighter color. And we can compare our model to data, and we can try to fit these effects of different mutations that best reproduce the evolutionary dynamics that we actually observe in data. So that's the main idea. And I'm not going to get too technical into this, but I want to give you a little bit of a kind of background idea of the methods and sort of what it is exactly that we're doing, and then I'll try to give you some intuition about why this might be a reasonable approach and how to understand the estimate that we can get out. So the idea is we have sort of a typical Bayesian approach where we're trying to maximize the probability of the model given the data. And we also have a prior distribution for our model parameters to help control spurious inferences. So in our case, the data is the distribution of variants, or in other words, genotypes over time. And the model here, we're talking about these fitness parameters, these sort of selection coefficients for individual mutations that go into this branching process. So it turns out in this case that you can exactly compute the posterior, but you can't really maximize it analytically. It's a little bit difficult to work with. So what you can do is you can make a simplifying assumption. So you can assume that there are a large number of infected individuals, so we're calling that N, and that the selection coefficients, the effects of any individual mutation, are relatively small. And that allows you to simplify the model and to actually solve it analytically. But the intermediate expressions that you get are not necessarily intuitive. And I'll tell you a little bit more about this in just a moment. But I want to focus on one part here, this limit that I'm not going to tell you about, but just to tell you by analogy with other things. So for people who are familiar with the diffusion approximation in population genetics, this is basically the same thing as that. Or in a more kind of abstract context, this is like the same thing as if I have a random walk on a lattice, I can take a limit and I get Brownian motion, sort of continuous time, continuous space Brownian motion. This is the same kind of limit there. So it's taking a discrete process and then turning it into a sort of simplified continuous time process. Okay. So we can write down this model for the likelihood of these distribution of variant genotypes over time as a function of selection coefficients. And it turns out you can actually analytically estimate the selection coefficients, the effects of these mutations that best reproduce your data. But the kind of intermediate expressions that you get are not necessarily intuitive. However, what you can do also is you can try to project the dynamics for variants onto dynamics for individual mutations. And in principle there, you actually lose information because now we're not thinking about like higher order correlations between different mutations. You're just looking at frequencies of individual mutations. But the intermediate expressions that you get are a little bit more intuitive. And interestingly, it turns out that this estimate that you get from the projection and looking at the kind of full variant level model actually give you exactly the same result, which is not obvious, but if you're interested, you can read more about that here. I'm happy to answer questions about that as well. But given that this is the case, what I would like to do is essentially show you the intuition that you get from this kind of projection and looking at dynamics of individual mutation frequencies, knowing that the answer that you get for this is the same thing as you get if you look at this whole dynamics of at the genotype level.
And could you clarify a bit more what you mean by projecting the variant dynamics onto individual mutations? Does it mean that you take, so you've got those curves that you showed, where in this case those curves would be for the different variants over time?
Yeah, so previously, and so the curves that I showed here
were for
this
mutations.
is actually so this is actually already sort of like at the mutation level. Yeah. So rather than at the level of full genotypes or something like this. And what we can do is we can write down, we can take these dynamics. So the branching process is really happening at the level of genotypes or variants. And then I can also say, based on the distribution of variants that I have in the population in a particular time, how do I also expect the frequencies of individual mutations to change? And that is, the expressions that you get to describe that are a little bit more intuitive. And so I know that this was a lot of text, and so I also have something kind of graphical to give you the idea. This is really what we're doing. So we have some model that's telling us how the distribution of genotypes is changing according to this branching process. It's driven by differences in fitness. And it's feasible to actually maximize this, but the expressions you get are not so intuitive. And what you can do instead is you can kind of project down from genotypes to look at individual alleles. So if I were to look at, and hopefully, let's see, can you see my mouse pointer? Yes, okay, fantastic.
Yes.
So I could say that, for each one of these individual genotypes, I might have different mutations in different places. And then here, sort of schematically, what I'm imagining is that in each row, I'm seeing like whether each one of these individuals here, each one of these variants here, has or does not have a particular mutation. And so in other words, I can look at mutation frequencies and how those are changing over time. I can look at the collection, distribution across all the mutations and say, in this sort of projected process, what's the likelihood of this given the selection coefficients? And in principle, this involves losing information because I'm not thinking necessarily, if I think about this, variance is like a collection of different mutations rather than also looking at correlations between them, I'm losing some information, but it turns out that the estimate that you get from this is actually exactly the same as what you get with looking at the genotype level in this simple case where I have an additive model for fitness. So if I had a more complicated model, then I will need to consider higher order correlations. But if my model is only additive, then I don't need to include that higher order information. Okay, so let me give you...
Sarah, go ahead. Sorry, John, maybe you can't see the raised hand, but...
okay, yes, sorry, no, I can't see the raised hand, but yes.
It's okay, and John, it's still your prerogative to punt questions to the end if you would prefer that.
No, it's fine. Yeah, happy to be interrupted.
Okay, well, this is a much more mundane question. What you showed here is really cool. What, and I don't know how I'd answer this in your shoes exactly, but what kind of, what are the most serious concerns with respect to data quality? And I mean, sampling evenness, representativeness, that sort of thing. And if we want to pump that again, that's fine. But that's what I'm thinking about when you show this. It's very cool. And I can imagine it, for SARS-CoV-2, but then it starts getting a little, shaky in other contexts.
Yeah, so this is a great question. And this is something that we think a lot about as well, how to deal with finite sampling in the data. And I'll talk a little bit about that a little bit further on. So I can tell you just, at the moment right now, what we do for our influenza analysis is we do some kind of smoothing the frequencies over time. And we smooth those compared to what we would do for something like SARS-CoV-2. We smooth them like fairly aggressively, I guess I would say. So we really try to kind of flatten this out more than we would with something like SARS-CoV-2, where it's sampled much deeper and with a kind of tighter temporal resolution. So that helps us deal with a little bit of this issue. We would really love to do like more geographical subdivision of the data, which we have actually done in our analysis of SARS-CoV-2. But what I'll show you for flu, we're just assuming that everything is, we're kind of mixing together the entire world, which we know is wrong. Like we do this understanding that this is not a complete picture. And so what I hope to show you today is sort of like a first result, making some approximations that we know are not completely, not completely reasonable, but this is sort of what you can get with simple approximations, and we hope to do better and be more, be able to break down the data in more kind of comprehensive and realistic ways as we go forward in the future.
We have another question from Marcus.
Oh, yes.
Just a quick clarification. So you mentioned that the effects of the mutations are additive. I assume that they're also type invariant, right? It doesn't depend on how many people have seen that infection or that mutation before in infections?
Yes, that's right. So for what I'm assuming right now, we're going to assume that these effects are invariant in time. This is an assumption that can also be relaxed. And so I won't actually talk about that today, but I'll just sort of briefly refer to some things where we have relaxed this assumption. We haven't actually done it yet for the case of flu. We've looked at that instead in the case of in HIV, but I think it would be super interesting to look at this for flu as well. But yeah, here we're taking sort of the simplest possible model that we can write down. Okay, so let me give you the intuition for sort of what our estimate gives and why this could make sense, at least at like conceptual hand wavy type of level. So let's imagine that we're looking at the frequencies of some mutations over time. Then based on our model, it would tell us, how I expect these frequencies to change in the next moment of time. And if I look at the frequency of an individual mutation, that change is going to depend on the difference in fitness between variants that have the mutation and the average fitness of the population. So in other words, mathematically, it's going to look something like this. And the idea here is that if the variants that have the particular mutation that I'm looking at, call it mutation I, if the variants that have that mutation have higher fitness than the average fitness of the population, then I ought to see them increase in frequency. And if it's lower, then I ought to see them decrease in frequency. And then, of course, there's going to be some noise, some kind of stochasticity on top of this. So I can expand this out, and I can look at the dependence specifically on the sequence. And what you get when you do that is something that looks like this. So here I'm going to show you like a schematic representation of the sequence as a guide with different mutations in different places. And so I have basically two contributions from fitness to how the frequency is changing. So one contribution comes from the mutation itself. So if this mutation I is beneficial, or in other words, if that selection coefficient is positive, then that's going to lead to assume that it's probably going to increase in frequency. If it's negative or deleterious, I would expect it to decrease in frequency. So that's the effect of that mutation. But then I also have an effect from the genetic background. So whether a particular mutation is going to be successful or not depends also on the background which it arises. So that's the second term over here. And to interpret this for you, this Xij is the frequency of variants that have mutations at both sites I and j together. And this is compared to the frequency of mutations at site I and site j. And taking the product of those two, that would be the frequency if these mutations were completely independent. So if, for example, like these two mutations I and j over here, if I have two mutations that appear together on the same background more often than I expect by chance, then that means their fates are going to be sort of linked together in some way. Right? So if, for example, let's say that this mutation I was completely neutral, it has no effect on fitness at all. If mutation J is beneficial, then I could still expect this mutation to increase in frequency just because it happens to appear together on the same background with that mutation J. And of course, this also happens, this correlation can be negative too. So it also depends, not only what mutations do you have on the same background as the mutation you're looking at, but also which ones are sort of competing with that mutation too. So what you can do then is you can say like, all right, I have this expression that's going to tell me how frequencies should change over time. And what I can get is a regression-like estimate of the fitness effects of mutations that are most likely to reproduce the changes that I actually observe in the data. So in the data, I can see how the frequencies are changing, and I know what those frequencies are. I also know what those correlations are. And then the thing that I want to estimate is I want to estimate these selection coefficients for these effects for the individual mutations. And that expression we can actually just write down. And here's the kind of intuition about that. So to kind of explain what this estimator is doing and how to think about it intuitively, let's imagine I have two different mutations here. And we can imagine that these are like in different populations. I've showed them on the same graph so that we can kind of see them together, but let's imagine that they're in totally distinct populations, and let's ignore the rest of the genetic backgrounds. So I have one in red and one in gray over here. So what our estimate would say is that if you want to get the selection coefficient for each one of these mutations, you should multiply their change in frequency over the entire time that I've been observing them with the inverse of the mutant frequency covariance matrix, which is the same thing as the linkage disequilibrium matrix for those who are familiar with population genetics, integrated over time. So this might sound a little bit confusing at first, but I promise that there's an intuitive kind of explanation for this. So let me go through the different parts. So the first part I have is the change in frequency. So if the change in frequency is positive, then, for example, that's an indication that that mutation could be beneficial. And the more positive it is, the more beneficial it might be. In this case, that doesn't really distinguish between these two variants or these two mutations because both of them are increasing from a frequency of near 0 to something that's near 1. So that's quite similar for those two. So the second part is this covariance that's integrated over time. And I think one way to think about this intuitively is to think about the diagonal part of this covariance, which is the variance. So that looks like frequency times 1 minus frequency. And intuitively, this is large when I'm at intermediate frequencies, and it's small when I'm near the boundaries, so near a frequency of 0 or 1. So if I were to take that and integrate it over time, roughly that's measuring for me how much time am I spending at intermediate frequencies rather than being like at the boundaries, which is either the mutation is not really there in the population or it's fixed or very close to fixed. And so if I did that for these two mutations, I would find an integrated variance that's much, much smaller for the red one than it is for the gray one, because it sort of shoots up very quickly from low frequencies to high frequencies. And since that's in the denominator, that would tell me that the selection coefficient for the red mutation should be a lot larger than the gray one. And this is sort of intuitive. If I have a mutation that's really beneficial, it ought to sweep through the population much faster than one that's sort of maybe modestly beneficial that might take a longer time to actually fix. So that's the sort of intuitive picture. What about the off-diagonal part? So these covariance terms are accounting for LD, correlations. So those are essentially measuring for you how much of the success or failure of a particular mutation is due to its individual effect versus the background that it appears to be on. So sort of disentangling these different terms. And what's interesting is that you might imagine, even in this very simple model, right? Even when I have this like additive model, that I might need, all orders of information. I might need to know about the whole variant distribution to try to estimate these individual effects. But it turns out that this is actually not true in this case, at least in the limit that these selection coefficients are small and then population size is large. In this limit, you actually don't need to know the higher order correlations. All you need to know is the frequencies of individual mutations and how they're correlated with each other. And that's all the information that you can get to determine or to estimate these effects of individual mutations. So this is a lot of hand waving. Let's look at simulations to see how this actually works. And here, I'm showing you a simulation that's a little bit different than the branching process simulation that I described earlier. This is more of a Wright-Fisher model type of simulation. It's common in population genetics, but the estimator that you get out is very, very similar up to a multiplying factor. So the intuition here is quite similar. And here I've simulated the evolution of a population that has some mixture of beneficial, neutral, and deleterious mutations. And you can see the mutation frequencies over time over here. And I would argue that you can probably guess that these ones that end at high frequencies are likely to be beneficial. The ones that end at low frequencies are likely to be deleterious. The ones that are in the middle are maybe a little bit harder to say. And that it might be difficult to just sort of pick out by eye which ones of these mutations are going to be beneficial and which ones aren't. But this is something that we can estimate. So if we apply this approach to this type of data, we can, for example, estimate or pull out which mutations are beneficial, and the trajectories that you see on the left-hand side sort of match with what would probably be your intuition, that beneficial mutations are going to tend to either start at low frequencies and go to high frequencies, or maintain at high frequencies for most of the time. Similarly, or conversely, for deleterious mutations, you can also pick out these tend to go from high frequencies to low frequencies or stay at low frequencies for a longer time. But what's interesting is that when you look at this information about correlations, you can also pick out these mutations that are really neutral. And these are not so obvious because some of the trajectories that you would see on the left-hand side over here can look very much like either a beneficial or a deleterious mutation. But in this case, what's really driving the dynamics of the frequency are not the effect of that individual mutation, but the effect of the sequence background. And we know that when we have the correlations, we can we can sort these things out. So since this is sort of a longer talk and has some more methods related things over there, I want to give you one more kind of piece of intuition, looking at a really kind of simple case where we can use this correlational information to disentangle things. So this is a particular very, very simple setting that we've engineered in simulations, where I imagine that I start with I have just two different sites in my sequence, and I can either have a mutant or not at each one of those sites. And I start with a mixture of individuals that have no mutations, and then ones that have a mutation just at the second site. And here, the mutation at site 2 is not beneficial at all. It's neutral, so it has no effect on fitness, but mutation at site 1 is beneficial. And what happens in this simulation is that I have a mutation at site 1 that arises on this background that already has this mutation at site 2, and then that ultimately sweeps in the simulation. So if we were to, and here I can show you, so this is what I was just saying. And then if I look at the frequencies of these individual mutations, so this is sequences that have mutations at site 2. This is sequences that have mutations at site 1. They look like this. So I have some of these sequences that have mutations at site 2 early on over here, and then it becomes, it overlaps completely with these ones that have mutations at site 1 because those go on to take over in the population. So if I were to look at the frequency of mutations at site 1 and mutations at site 2 individually, and I were to ignore this kind of correlation between them, then if I try to estimate how beneficial are these mutations, I would estimate both of them to actually be beneficial, even though this one at site 2 is really neutral. And so the distribution that you see over here is a distribution over simulations that are with similar conditions. However, when we take into account the fact that we have this correlation, we can properly disentangle these two things. So of course there's noise in these estimates because these are real simulations, there's stochasticity, but you can separate out which mutation is actually beneficial from the one that appears to be neutral. And so this correlation information is really giving you quite a lot of power to discriminate between the effects of different mutations. Okay, so what I'll tell you about is our application of this to flu, and what kind of encouraged us to look at flu is encouraging results that we had when we looked at SARS-CoV-2. Just want to briefly mention why we thought this was interesting and worth continuing to develop. So we looked at global data from SARS-CoV-2 collected across many different regions, and this is from the beginning of the pandemic in 2020 through February 2024. And so we could use this approach to try to identify different mutations that might affect the transmission of the virus. And in this case, we did this genome-wide. So the dots that you see here are effects of individual mutations that we infer. And the bigger that dot is, the larger the effect. The ones that are in red are beneficial ones for the virus, so we would say that there's increased transmission. The ones that are in blue are deleterious or should be harmful to transmission. And these different tracks that you see here across the genome in a circle are the different mutations or different variants at the nucleotide level here. So one of the things that you probably see really immediately when you look at this data is if I look at, where are all the big dots, you see that those are in spike, or at least a lot of them are in spike, which is nice to see. So this model doesn't actually know anything about where is spike and where are these other proteins. It's just based on the sequence data alone with no other, phenotypic information or anything like that comes into the model. And we see a lot of selection for mutations that make a lot of sense based on the incredible amount of experimental work that's been done to try to characterize all the different variety of mutations that we've seen in SARS-CoV-2. One thing that I think is really interesting about this, though, and why it's nice to have a statistical approach that's not really directed, let's say, solely at spike, where we know it's going to be biologically important, but is just more generally looking at what are genetic changes that appear to be associated with driving changes in transmission, is that sometimes you can be surprised and you can pick up things that wouldn't have been your first guess. So actually, if we look at what mutation appears to be, which are the top mutations that we would say increase transmission the most. Okay, so sorry, this I skipped. I also have mutations that are outside of spike that are interesting. But if I look at those top mutations, one of the mutations that we infer to really consistently be within the top 1 or 2 is this mutation in SP4. So not the first place that we would tend to look at. And actually, for a while, when we were preparing this work and when it was when we were going through revisions, we didn't really understand why this was a mutation that was inferred to be really beneficial. And so the kind of experimental characterization for this only came later. And interestingly, what was found is that this mutation is very close to a cleavage site between an SP4 and an SP5. And it helps to enhance the efficiency of cleavage at this boundary between an SP4 and an SP5. So it leads to increased production of these non-structural proteins, which has effects downstream on antagonizing the immune system and also on viral replication. So it appears that there's really something biological that's underlying this. And I think that, this is something that can be nice about this approach, that when you just apply it to the sequence data, you can be pointing to directions that, might not be the first that you would look at. All right, but enough about SARS-CoV-2.
Actually, sorry.
One
question going back to SARS-CoV-2
yes.
Before we leave it. Yeah, if you go back to the plot before.
Yes,
of course. So obviously you're not seeing very many deleterious mutations because these are mutations that would have had to accumulate sufficiently in your population to be able to measure anything at all.
Right.
And so did you find that for the ones that you can measure, is it because they're linked to positive mutations that they happen at all?
That's a great question. So you do certainly see those as part of the kind of major variants. We have not looked in a lot of detail to try to sort out the kind of, or to understand the deleterious variants specifically that we infer. Part of that is that it's quite hard to validate, I guess. There aren't often a lot of experiments that are kind of saying like, okay, this is a mutation that's really harmful to the virus in some way. And so it wasn't so obvious for us how to validate these extensively as we could for the mutations that appear to have some sort of beneficial effects, ones that are affecting antibody escape or things like that. That was much easier to check and validate. But yeah, these certainly appear there on the background of some of the major variants. Some of those are lost. Some of them might only appear sporadically and never really reach high frequencies. But we haven't characterized those very comprehensively. I think it would be interesting at some point to go back and try to look at this more carefully. But for us, they were a little bit more difficult to validate than the ones that were, than the sort of beneficial mutations. Yeah, and we see a sort of similar pattern to foreshadow in flu as well, where you see more of the beneficial mutations than the deleterious ones, and this is probably because mutations that are significantly deleterious, you're just very, very unlikely to observe them, right? Okay, so I wanted to comment briefly on some of the other methods that people commonly use to try to estimate fitness of either different variants, or to try to do predictive modeling of either dynamics of flu or other pathogens. So one of these methods is multinomial logistic regression. So here I've just given one recent reference for this. There are many, of course, that have applied this approach. And this is a really popular approach for estimating variant fitness. So I wanted to sort of contrast our approach versus this approach to kind of give an idea of differences in assumptions and things like this. So this MLR approach, it incorporates sampling noise. So the fact that, at some particular moment in time, I might only have 10 sequences that are sampled, but at another time I might have 100. So that's something that's incorporated into this model. But in this model, you don't have drift. This sort of assumes that the underlying dynamics are actually deterministic. So ours sort of has the opposite incorporation of stochasticity. So we have drift in our model, but we don't have sampling noise. So that means that we need to process the trajectories. We need to sort of smooth trajectories if they're poorly sampled to make sure that we're getting results that are reasonable. So we're also working on doing joint inference of underlying frequencies along with effects of mutations. And this is something that is tractable. It becomes more complicated, but it's something that can be approached as well. And look forward to hopefully next year sometime this will be out somewhere. But that's something that can be done, and that's one difference between these approaches. I think that maybe the major difference between them is sort of let's say the default fitness assumptions. So at least by default, in this MLR, you typically assume that each sequence that's kind of clustered into particular variants or lineages is going to have the same fitness, and that there's not really an a priori relationship between the fitness of different variants. And in our case, with this sort of default or simplest model, the simple additive model, we try to explain differences in variant fitness based on the effects of individual mutations. So that if I have sequences that are similar, then they probably should have similar fitnesses in our model, modulo the effect of really large effect mutations. And this is how you really get large effect mutations out of the model, where small genetic differences result in big differences in growth. Our fitness model, though, can go beyond this, so we don't necessarily have to have a purely additive model. We can include epistasis, for example. We can include more complicated metrics. We can also include fitness effects of mutations that are time varying, so that's this last one over here. I won't talk about any of those today, but just to point to, this is something that potentially can be extended. And of course, you could also do the same thing for MLR, right? You could also have some kind of, let's say, a prior that's going to relate the fitnesses of related variants or something like this. So certainly these models or these approaches for modeling the data could be extended in different ways, but this is just sort of one attempt to kind of summarize some differences between the approach that we're taking and ones that are common, I guess, right now. All right.
And so are you going to show us if they differ in their results?
That's a good question. So I have not applied those. We haven't applied the MLR approach to data here, and I think this would be something that is really interesting to explore. I think maybe, if someone would like to work on this, please send us a message. I would love to see it, because I think there's also some kind of you have to make some choices about what you define as a variant, right? And that, I think, is, that's the type of analysis that we have not done before. And so I wouldn't want to make bad choices because we haven't done this very much, and then get, poor performance and then say like, I think it would be really interesting to look at. In this case, I'm not going to show a comparison between different things, but we would be super interested to explore.
There's another question from Sarah.
Oh, yes.
Yeah. So again, possibly for later, but a major direction that multiple groups here and just the world, is moving in is incorporation for something like flu, SARS-CoV-2, fitness estimates derived from like human sera.
Yes.
Then obviously deep mutational scanning data. And so I'm wondering if you've thought about the incorporation of those data types in particular.
Yes, I have. I've thought quite a bit about this. We have some ideas for how we would like to do this, and have even written up sort of a proposal for how we would plan to do this. So I think, this is a great way to get my caveats out of the way before I do the flu analysis, where I would say that, okay, we've made a lot of simplifications here. We assume that fitness is additive and it doesn't change in time, and we haven't thought about antigenic differences or anything like this. So this is not to say that we think that those things are not important, but this is sort of like the simplest model that we can write down, and it can potentially be extended in different ways. So if you know, for example, that a particular mutation has an antigenic effect, then that's something that you can incorporate in your model in different ways, perhaps through something like a different prior distribution. I think there are multiple ways to incorporate this, and I'm actually super excited about doing this in the future. What I hope to show today is that even with simple assumptions, you can actually get some interesting results, and then hopefully we can actually improve this and do much better in the future. Yeah, so great. So let me go on to talk a little bit then about flu and how we've actually applied this to real data. And this is the kind of picture that we had before, right? So thinking very generally, we have this drift that we know is driving evolution of these viruses, where this is progressive immune escape. But it's turned out to be quite difficult actually to predict flu evolution in a quantitative way. And I guess it depends on how you define difficult, right? But it's been challenging to make predictions with really 100% accuracy, and we certainly are not going to approach that either. But it's more challenging, let's say, than one might expect. And I think one way that is really kind of a dramatic example of this is trying to predict whether individual mutations will fix or be lost. So this was studied by Barat-Charlet and collaborators a few years ago, trying to use a really excellent phylogenetic predictor called LBI to try to identify, if I look at mutations that are at some particular frequency in hemagglutinin, which ones are the ones that are more likely to fix, and which ones are the ones that are more likely to be lost. And that's basically what this figure is showing you here. It's taken from this paper. So the horizontal axis is the current frequency of the mutation, and the vertical axis is the probability or the fraction of these different mutations that fix. And the dots that you see in blue here is just all the mutations in the data set. The ones in this orange are ones that have high LBI, so you would predict, based on this phylogenetic predictor, that they are more likely to fix. The ones in green, you would say that they're less likely to fix, but actually these all look fairly similar to each other. And it's really quite hard to distinguish which mutations are likely to fix and which ones are not. These also lie quite close to the expectations that you would get just from a neutral model, where mutations have no effect on fitness at all. And in that case, the probability of a mutation fixing is just equal to its frequency. So it's just kind of chosen at random which mutations will fix and which ones won't. So this is the sort of, this is one of the things that we wanted to try to test. Could we use our approach to make better predictions about which mutations will fix and which ones will be lost? And can we also predict how the composition of the viral population might change over time? So what we did is we analyzed data from GISAID from January 1st of 2009 through early 2020, the onset of the pandemic, to estimate the effects of mutations on flu transmission. So now looking at the amino acid level rather than the nucleotide level. And we looked at hemagglutinin and neuraminidase for both H3N2 and H1N1. And for this initial analysis, we looked at them separately because we found that it was more difficult to get like whole genome sequences of flu. If we look for that, we find very, very few sequences. And so the data was a little bit more difficult to deal with. In this case, we get about 50,000 sequences for each protein and for each subtype that we're considering independently. And we estimated these fitness effects for each protein and each subtype using just the worldwide population dynamics. So not trying to do some geographic reweighting or something like this, which I think in principle would be like a smart thing to do, but just taking the sampling that we have and using that to compute frequencies over time and using that to try to estimate the effects of different mutations. So here's what we found. This is the frequency of different mutations for H3 and N2 and for H1 and N1 based on this data for this time range where I'm omitting or not plotting mutations that were already fixed at the initial time point over here. And on the right-hand side, I'll show you our estimates of fitness effects for these different proteins. So this is what we get for H3 and for N2, and this is for H1 and N1. And I was really struck when we got these results by how similar these distributions look across these different proteins. So in each case, we have, oops, I have to click my mouse again. In each case, we have a peak that's near zero or neutral. It's a really large peak. And I should say that since our model incorporates regularization, this is not to say that most mutations are actually neutral, that really we're sure that their effect is zero, but rather there are a lot of mutations that are at fairly low frequencies where it's not obvious that they have any non-zero effect, either beneficial or deleterious. And so their most likely effect gets collapsed near zero. So a lot of mutations are in this category. I have kind of a long tail of rare beneficial mutations on one side, and on the other side, I have a much sharper tail that falls off on the left side towards these mutations that appear to be substantially deleterious, that we would estimate to be substantially deleterious. So that's what we find when we look at the data, but how can we understand or how can we think about whether these are reasonable inferences or not? So what we tried to do is we tried to look for mutations that have known functional effects that are important that we could try to compare with, or known like functional regions. And so this is looking at hemagglutinin, and particularly looking at the receptor binding site. So we know that this is an area that's important for cell entry and also for antigenic change. So there have been a lot of papers that have studied this, and I'm just going to highlight one over here as an example. So this is from Cole et al. From a little bit more than 10 years ago now. And what they did is they tested the ability of different sera to neutralize H3N2 variants over a long stretch of time. And they found that mutations near the receptor binding site were ones that would commonly drive antigenic change. So here, this is a zoomed in view of this part of hemagglutinin. So the receptor binding site is in yellow, and these major antigenic change mutations are in red. And what we can do is we can look at the distribution of selection coefficients that we see for all mutations in hemagglutinin at other sites other than these. We can compare those to, for example, the distribution of selection coefficients that we see for mutations that are in the receptor binding site. Excuse me. So when we look at this distribution, here's what we see for H3. So in gray, these are mutations at sites other than the receptor binding site. And the ones that are colored in here are mutations that are in the receptor binding site. And this is a log scale for frequency over here. So these are quite rare. Most of them are in this area. But you can see I have a longer tail towards mutations that have positive effects for ones that are in the receptor binding site. You can do the same thing also for H1. Distribution looks like this. And this is something that, if you want to, we can do statistical tests to see if this is really a significant difference in distribution. And in this case, we find a pretty significant difference in these two distributions. There are much, much fewer mutations that are in the receptor binding site. But when you see mutations there, they tend to have larger effects. And those effects also tend to be more positive. We can also look at these specific antigenic sites. So here for H3, I'm looking at the colloidal site, which is really a small number of sites. It's only like 7 sites. And you can see that those are also highly enriched in beneficial mutations. For H1, we referred back to this much older analysis from Kim et al. In the 1980s that identified a much longer or a much bigger set of antigenic mutations. So it's not nearly, I guess, as kind of precisely targeted as the colloidal work, but you can still see a significant difference in distribution between mutations that lie in those sites versus ones that don't. So we tend to see that they have an enrichment in beneficial mutations that are either in the receptor binding site, which we know to be functionally important, or in previously identified antigenic sites, which are sites that could be affecting antibody responses. And so this sort of makes sense in terms of whether or not these effects that we're inferring are maybe capturing something about features that could be important for viral transmission. So what we wanted to do then is we wanted to take this and see if we can use this information to actually make predictions. And so what we do is we have our branching process model that I'm using I'm calling this out here sort of in jest, but the branching process model is a generative model, if you like, that we can actually use to run simulations, or as we used to call these models before the last few years of AI, it's a model, right? And so I can use this to actually estimate for me how the population might change over time. And the way that we do that is we imagine, let's say we're going to make this prediction at this time t here, like t is equal to 200 in like arbitrary units. So I look at the dynamics up until that time. And then what we would do is we would get a distribution for these selection coefficients. So we would infer our most likely estimate to the selection coefficients, and then also some uncertainty around this. And then we can sample from that distribution and perform a lot of simulations to see how the population might evolve in the future. And if I want to estimate which mutations are going to fix and which ones will be lost, I can run these simulations many times, and I can rank different mutations by the fraction of simulations in which they fix, and compare those to a null model where the probability of fixation is just equal to the frequency of the mutation. And the results that I'm going to show you here are from 2012 through 2020. So we start inferring parameters in 2009, and we need a little bit of data to actually get parameters in the first place. So we start, immediately on January 1st, we have no idea what the effects of all these mutations are. So the predictions very, very early on are not so good. But then as you get more data, you have more information to actually tell apart the different mutations. So first, what I want to do is show you this by looking at AUROCs. So this is like a classification metric for how well you're able to divide mutations that will fix versus ones that will be lost. So random guessing would be an AUROC of 0.5, and perfect classification would be 1. So first I'm going to show you the null model. So interestingly, the null model actually does a little bit better than random. And this is because, let's see, when you bend mutations into frequencies, right? So I can get a distribution. I have some mutations that are going to be on kind of the low end of the frequency bend, and then some mutations that are going to be on sort of the high end of the frequency bend. So that actually does give you a little bit of predictive power. It makes it on average a little bit better than random, but not much better, as you can see. And if we look at our simulations, then we get something that looks like this. So we can actually improve quite a bit over null model in terms of predicting which mutations are likely to fix and which ones are likely to be lost. So if you look at, the AUROC, you can see that it's definitely not 1, right? So we can't make these predictions perfectly, but still we can do substantially better than what you would expect by chance in these simulations. And I'll show you this in sort of a different way as well. So these are plots that look a little bit more similar to the plots that you saw in the Broad-Charlet et al. Paper. So this is grouping the mutations into different categories. So here, the ones in this dark red are the ones we would say are the most likely to fix the top quarter within each bend. The lighter color is the top half. The gray is all mutations, and then the light blue is the bottom half, and then the darker blue is the bottom quarter, the ones we would say are really the least likely to fix. So these are our predictions for H3 and for H1, so the 2 hemagglutinin. And one pattern that we see pretty consistently is that as I go to either really low or really high frequencies, it's often a little bit difficult to distinguish between mutations, but we have a lot of discriminative power when we're in the middle. So those are what we get for the hemagglutinins. If I look at neuraminidase, this is what I have for N2, and this is what I have for N1. So N1 really looks fantastic. In this case, we're doing really, really well there. And that one, I think, is one of the ones where we're sort of the most successful in this case. So we can really we have some ability through these simulations to kind of point to mutations we think are more likely to fix and ones that we think are less likely. I'll return in a little bit to talk about why it is that we think that that's the case and where we think that this power is coming from. Before I get into that, I want to talk about making predictions of the composition of the viral population in the future. So in this case, what we're going to do is what I'm going to show you is estimates of the viral population 9 months into the future, which is sort of very roughly a time scale that one might have for vaccine design. And of course, the vaccine design question, as you all on the call here know, it's a very, very complicated question, and there's lots of factors that are involved other than just saying what the viruses might look like in the future. But this is at least setting a time scale that we might be interested in for this type of problem. So what we do is very similar to what I was mentioning before for the predictions of which mutations we'll fix and which ones we lost. So we run a lot of, I didn't describe it here. Okay, we run a lot of simulations, basically, and then what we found is that if you look at the typical simulation that comes out, the typical simulation looks pretty close, or is a good representation of the future sequence distribution. But how do you know what is a typical simulation and what's not when you don't have access to the future distribution? So what we do to get this estimate is we run a bunch of simulations, and then we measure the distance between the simulated populations in the future using this Earth movers distance. So it's like a weighted Hamming distance, also a Wasserstein distance metric for those who are familiar with that terminology. And what we do is we pick what's called the metoid to kind of characterize like a closest to central population as our like predicted population for what the future will look like. And so we can compare that to what actually happens in the future and to a null prediction, where again, a sort of null or neutral prediction would be that the population in the future is just going to be exactly the same as what it is when you make that prediction, you know, assuming that everything is neutral. So here is this plot showing on the horizontal axis the time that we're making the prediction. So the measurement comes 9 months after that. We're trying to get 9 months after that what the population looks like. And this is showing improvements in the model prediction versus what we get for the neutral model. And so if I'm above this dotted line in the background, then I'm doing better than neutral. And if I'm below that dotted line, then it means I'm going to do worse than neutral. And we make these predictions every month throughout this time. So here's what I get for H3N2, hemagglutinin. So a lot of the time I'm spending above this line, though I do have some case over here that doesn't do so well. More about that in a little bit. This is what we get for N2, also improving over the null quite a bit in most cases. We have also H1 here, which is almost always better than the null distribution, and then similarly also for N1, which I think maybe is only at one point it's getting close to the null. So we really are typically able to produce predictions that are going to get you a better estimate of what the viral population might look like in the future than sort of null expectation. And I had, of course, this point over here where I did really poorly. And in fact, Sarah was the person that pointed to why this is when we were, when we were meeting sometime in the past when Sarah was visiting over here. We realized that, in fact, there was a reassortment event that happened at this time, where you had a sort of dying clade of hemagglutinin that fell off, was on its way to becoming extinct. Then through a reassortment event, this hemagglutinin actually got put onto a different sequence background and ultimately surged to become dominant in mid-2018. And this really coincides exactly with this time point over here where we have predictions that don't work so well. And if we were to remove that specific clade from our predictions, we can see that this value goes, right back up to neutral or a little bit better than neutral. So it's really specifically this clade where we make predictions that don't work so well. So we can even, typically we're able to improve upon the null. And in these cases where we do significantly worse, we can identify, at least here, why it is that this is the case, or what's a likely explanation for this. So I wanted to kind of close here, and I think, okay, great. Doing good on time. So I wanted to close here with kind of why it is that we think that we get some predictive power from this approach, or what's important for predictive power. And one of the things that really emerges as something that is important is accounting for the sequence background. And I'll show you kind of one example here, and then in the next slide, some very preliminary work. Where this is looking at, focus just on H3, but is general for the other proteins as well. So I can look at my model and I can kind of take different features out of it and see how well that performs in making predictions. In this case, making predictions about which mutations will fix and which ones will be lost. So this is what I get from the full model. And then if I remove from this LD when I'm trying to estimate the fitness effects of mutations, I do a little bit worse, especially in this kind of middle over here, but I'm still able to make some predictions that are okay. But when I also ignore LD in simulations, I still have some places that are improved relative to null, but now I'm really doing significantly worse. So taking into account the sequence background is something that appears to be important for predictive power. And we can look at that a different way too. So one of the things that we've been looking at very recently is to compare how the background fitness changes over the lifetime of a particular mutation. And what we do is, here I have two hypothetical mutations. So this is not from real data, but just from a simulation for illustration. So once they cross a minimum frequency threshold, which in our case is 5%, I'm going to compute the background fitness. So in other words, the average fitness of all sequences that have that mutation when it first crosses this frequency threshold in the population. And then what I'm going to do is I'm going to look when that mutation either fixes in the population or when it's lost, I'm going to compute the background fitness again, and I'm going to look at the difference between those two. And you might imagine that if the fates of individual mutations are driven entirely by their, the effects of that mutations, the effect of that mutation, then the background of mutations that fix and ones that are lost should be exactly, the same. It shouldn't be different between them if there's no hitchhiking or anything like this. But if I have background effects that are important, then the mutations that fix ought to gain more fitness from the sequence background than the ones that are lost. And indeed, if we look at this, we see that there's a difference here, and the statistics are a little bit more difficult to compare because I'm just looking at a fairly small number of mutations, putting some restrictions there on I want to look at mutations that exist in the population for at least a minimum amount of time. Otherwise, I'm looking at ones that sometimes are very, very low frequencies. So I can see a difference in these distributions where for the mutations that fix, so those are the ones in colors over here for the different proteins, those tend to gain more fitness than mutations that are lost. But of course, there's one easy explanation for this, which is that mutations that fix are probably in the population longer than mutations that are lost, right? So mutations that fix, they have to go all the way from a low frequency to a high frequency, whereas ones that are lost, they could just stay at frequencies, at 5% or a little bit above, and then drop back down below. And so they might not be in the population for such a long time. So if I'm just accumulating mutations over time, then I could, I could see this difference in backgrounds or in the change in background fitness, even if there really isn't any difference in the distributions. So what I ought to do is I ought to normalize them by time, right? I ought to say, what's the rate of either increase or decrease in fitness of the background for mutations that fix versus ones that are lost, to see if mutations that ultimately fix are really getting a boost on the background or whether they're basically the same as the ones that are lost. And so when you make this correction, the distributions come a lot closer together, which makes sense, but you can still see differences between them that are significant, even though the sample size is not so large. So mutations that go on to fix tend to see a more rapid increase in fitness on the background than mutations that ultimately go on to be lost. And I think this is another way of pointing out that the sequence background is really important, which I think is also sort of connects with things that people have been looking at theoretically also recently in the context of modeling flow evolution. So let me come then to kind of summary and conclusions, which is that we can use these types of models to extract information from evolutionary data that can be complicated. When we applied this to IIV, we find mutations that are beneficial, that are in key functional regions, and we can also use those to make some predictions about future population evolution. And the sequence background appears to be especially important for these predictions, at least in our model. We can relax these assumptions. I think doing that and also incorporating more phenotypic information could potentially improve predictions. And I'm really excited about that, especially things like the effects of antigenic information, which people here have shown that this is important for being able to identify the lineages that are likely to be successful in future seasons. So we would love to incorporate that information as well. So let me thank the lab over here. And I realize now I forgot to mention in the beginning. So this work that I was showing you today is mostly the work of Edwin Rodriguez-Huerta, postdoc in my lab. And I thank my collaborators here and support that we've had in the past. And be happy to take any additional questions.
Thank you so much.
I have a question. No one else wants to start. So I'm trying to interpret your results in light of the Barré-Churlet paper, which I haven't read in a while. But if I remember correctly, their argument, right, was that because it seems that the best predictor of the fixation probability is the initial frequency at any given time point, this like effective neutrality that emerges. And if I remember correctly, their explanation is that the models that assume a fixed effect, a fixed fitness effect of any mutation, right, they fail to account for the fact that as immunity builds up, those fitness effects go away, and so you can't really model it that way. But then is it fair to say then that your results suggest an alternative explanation, which is even if you assume those fixed effects, as long as you account properly for the linkages equilibrium, as you were saying, then that can reconcile the observations with this?
Yeah, so I think, right, so this is a really interesting question, and a really interesting point to explore, and something we've been trying to understand as well. So I think that there are at least two possible components to this. So one is that so we know for sure that the fitness effects are not constant. That's for sure. It could be that the time scale over which they're changing is actually a little bit longer than one might think. And so even though on the long term, this constant fitness model isn't good, or we know that it's going to break down, on the short term, you can still make some reasonable predictions. So I think that's sort of one possible interpretation. Another interpretation that's there, especially in recent modeling that they've done, is, basically there's this kind of idea that you can have some mutations that are transiently beneficial, and they reach sort of intermediate frequencies, and then what happens after that then really just depends on the background. So do you get other good mutations that come on that background, or do they come on other backgrounds and then maybe those mutations die out? And in our analysis, we're finding that the background effect is really important. So maybe it's kind of a combination of different factors, that the time scale over which these things are changing might be, let's say, slightly longer than we might have imagined. And then also when you account for background effects, maybe that helps to also sort of paper over the lack of accuracy that you might have with assuming that fitness effects are constant, where if you have additional mutations that are coming on the background that are helping to drive you to fixation, you can include that information in your model, and it can help you to make predictions. So I think my personal explanation is a combination of these factors.
Great. Do we have any more questions?
Well, just a follow-up comment, John. I hope somebody is maybe if they watch the
recording stopped.
later. I was going to say, I hope somebody's excited about this comparison with MLR. I think it would be really nice to do some, just some tests, and then be really happy to talk with you further about incorporation of human, serological data. And
yeah, I mean, it's so important, right? I think that would be really, really interesting to do. And I think also, so on both fronts, right? I think the comparison with the MLR would be really interesting. And we can even sort of within our own framework as well, we could look at, because then it's sort of not an issue of like, well, is this like our hands or is it, a model related thing? We can look at a version of the model where different variants or genotypes have different fitness, which is also inferable in a very similar way. And that I think would be conceptually quite close to MLR, at least like vanilla MLR. So I think that would be something that would be very interesting to compare and to look at. I hope to get more of these things moving as we go along. But that's one of the things I'm excited about, for sure.
I assume we don't have any of the MLR people on the call, because I feel like they would have jumped in already. But just
sorry, they're at the strain selection meeting.
Exactly, that's right.
Oh.
Yeah. Unfortunately,
they have things to do. Well, you all have things to do too, and so of course, I'm very happy that you chose to be here.
But I think off the top of my head, from what we've seen with their projections, they do pretty well up to 6 months. But actually, I feel like after that it broke down. Like they didn't have results at 9 months, like John, that were so good.
Yeah, I mean it's very like I think one reason why I've also, let's say, kind of tried not to emphasize, I guess, the comparison with other methods is I know it can be tricky, right? And like what you choose for your metric, and for example, like how frequently you do these comparisons or things like that, those can all affect the result. What exactly is your data set, right? When your data was collected, and all of this can affect what results you get out on the other side. So one has to be careful, of course, with these comparisons. But no, I think in our case, if we look at let's see, yeah, and then we have I'm gonna skip ahead a little bit. Okay, yeah, if we look at distributions of improvement versus null, in some cases it looks quite nice, and we can go out also to a year, for example, and sometimes even do just a little bit better, which I think is mainly due to null getting worse rather than our predictions getting much better. But in some cases, like for example, for H3 or for H1, we can do on average like quite a bit better than the sort of null prediction, which I think compared to at least things in the Huddleston et al. Paper, which of course there's more things recently, compares quite well. So we would be really interested to look at a sort of comparison of approaches, for sure.
Although to correct myself, they are publishing the 12-month forecast, so I guess they're happy enough with them to publish the 12-month forecast for influenza variants.
Yeah.
Great. Well, if there are no more questions, actually we've come to the end of our time anyway. So thank you so much, John. That's so useful for our group and for everyone else who attended as well. Thank you. It was really fun. Our next speaker next month is Nick Rice. He'll be talking about SARS-CoV-2 nowcasts, and we'll be inviting people to that seminar soon, and I hope you can join us. So thanks again, John, and see everyone else in a month.